Welcome everyone to our journal club that we're starting in association with our PAMCA bioinformatics workshop. The purpose of this journal club is to highlight how genomic data can be applied to the questions that we brought up in the previous workshop. So we had talked about using genomic data analysis to look at target site resistance. And today, Dr Clarkson is going to talk about his paper, The Genetic Architecture of Target Site Resistance to Pyrethroid Insecticides in the African Malaria Vectors Anopheles gambiae and Anopheles coluzzii. Chris is a data scientist at the Wellcome Sanger Institute. So go ahead and get started, Chris.
Thanks, Brandy. Hi everyone. It just seemed like a really good time to do this journal club and talk through this particular paper, because so much of what we looked at last month in the workshop is based around these kind of analyses. And in fact, some of the analyses that we did in the workshop, you will see popped up here. Before we start, I just want to say, if you've got any questions pertaining to different bits of analysis that I'm talking about, or if you don't understand something that I've said, please feel free to speak up and ask a question during the journal club. For more general questions, there'll be time at the end to ask those. Let me share my screen. To say, it's way more intimidating to do a journal club on your own paper than it is to do one on someone else's paper, which is interesting. Also, we've got Alistair in the room. Alistair did some of these analyses. So Alistair, please feel free to jump in if you fancy explaining something. That would be very welcome. Let me share the right screen. Okie doke. So can everyone see that screen?
Yeah, you might want to just zoom in a little bit so things are filling up more of the screen. Yeah, that looks good.
Okay. Right, so we're going to look at this paper, The Genetic Architecture of Target Site Resistance to Pyrethroid Insecticides in African Malaria Vectors. We're looking at Anopheles gambiae and Anopheles coluzzii, because those were the species in the sample set that we are analyzing here. So let me just sort of talk you through the motivations and the broad strokes of this paper, and then I will get into the different analyses. Let me just zoom out a little bit so you can see the abstract there. Okay, so pyrethroid insecticides have been the cornerstone of malaria prevention in Africa for almost 2 decades. We know that all the bed nets, or currently all the bed nets, are using pyrethroids in some form, whether they're using them on their own or with some sort of partner chemical. Sometimes they're used in IRS, and there'll be widespread use of pyrethroids in agriculture. Resistance to these chemicals is very widespread and appears to be increasing. And that's a big concern, because we just don't know what's going to happen to our vector control plans with this increased pyrethroid insecticide resistance. And in fact, the WHO stated that we need to improve both our knowledge of the molecular mechanisms of resistance and our ability to monitor those in natural populations. And that's really the motivation behind this piece of research. We learned a bit about pyrethroid resistance in the last month's workshop. So we're looking at the voltage-gated sodium channel, which is the physiological target of pyrethroid insecticides. The pyrethroid molecules bind to this protein and basically force it open. It keeps it in its open state, and this causes paralysis, knockdown, and death for the mosquitoes. But amino acid substitutions, or non-synonymous mutations, which cause amino acid substitutions, alter the protein interaction with the insecticide molecules, and this increases the dose of insecticide that's required for knockdown. And so this is why we call this knockdown resistance, or KDR. So what did we do next? So we looked at the genome sequence data from phase 2 of the Anopheles gambiae 1,000 Genomes project. And so this is 1,142 wild-caught mosquitoes from 13 countries. And we looked at the genetic variation across the VGSC gene. And in addition to the known resistance alleles that we were aware of at this point in time that have been validated, which is 995F and 995S, and these are the KDR mutations, and N1570Y, which is an accessory to 995F and increases the resistance phenotype. On top of those, we described 20 other non-synonymous nucleotide substitutions at appreciable frequency in the VGSC. So these are SNPs that look interesting when it comes to pyrethroid resistance. And we mapped these onto the protein model to investigate the likelihood of pyrethroid resistance phenotypes. This raised a number of particular SNPs that looked very interesting and looked like that they warranted further research. Actually, that research has been done, and I'll get to that in a minute because it's quite exciting. We also analyzed the genetic backgrounds carrying resistance alleles, and this enables us to do a few things. It enables us to look for recent positive selection, and it enables us to track these haplotypes, these mutations, if you like, through space and time by using the genomic sequence around these resistant alleles. So it's not clear when, where, or how many times pyrethroid target site resistance has emerged. Geographical paths carrying resistance are not known. Some work has been done on this before, but never looking at the whole of the VGSC gene. So this research is the first to sort of really step back, look at the whole of the VGSC gene and the sort of surrounding genomic sequence that allows us to look into the history and the movement. So yeah, I think let's have a look at the results, and we'll talk through these. So this table, I'll need to do a bit of. Right, this table here might look a little bit familiar. So the first section of the results is VGSC non-synonymous nucleotide variation. So to identify variants with a potential functional role in pyrethroid resistance, we extracted single nucleotide polymorphisms that alter the amino acid sequence of the VGSC protein. So this is exactly what we did in module 4 of the first workshop. We looked for non-synonymous mutations, and we looked for those that were at or above a 5% frequency. And that's because we expect mutations that are under selection, you know, if they're causing some sort of insecticide resistance phenotype, that would be beneficial to the mosquito. We would expect those to rise in frequency. So we just look at those non-synonymous mutations over 5%. And then here we can see 23 variant alleles that fit this criteria. And it just looks very similar to those tables we produced in the workshop. We've got the position of the nucleotide change and then the nucleotide change itself. So here we've got, this is the position on the 2L chromosome arm, because that's where the VGSC gene is. We've got a G to A nucleotide change, which causes this R254K amino acid change. We've also put the position the amino acid position that changes in Musca domestica, which is the housefly. And that's because a lot of the research into VGSC and insecticide resistance has been done using the housefly. So a lot of people know these positions based on the housefly amino acid numbering. We used to call the KDR mutations 995S and F. We used to call those 1014F and S. And as you can see here, that's because in the Musca domestica genome or gene transcript, these are actually at the 1014 position in the gene. But actually, when you look at them in mosquitoes, they're in the 995 position. So it just makes it a little bit easier for us to use the numbering in the mosquito reference genome. So then we computed the allele frequencies of these SNPs among our 16 mosquito populations. And here we defined them by species and country of origin, which is a kind of quite a low resolution cohort. Because if you remember to the last workshop, when we looked at cohorts for our allele frequencies, then we split by country, by species, by administrative region, and by year of collection, which gave us a much higher resolution and perhaps more biologically interesting cohort. But here we've done a very high level, and we've just split them by country and by species. And we can see here are the cohort columns with our frequencies. Something that really struck me when I was looking at this table was how much easier it is to read or to interpret the new heat map tables that we were making in the last workshop. And actually, if we made this paper again, I think we'd use a nice heat map here, because it's quite difficult to see what's going on when it's just this sort of plain table. Okay, is there anything else I wanted to say about this? Okay, so then let's look at the next figure. I need to turn my page back the right way around. Okay. So we then mapped these 23 non-synonymous variants on a transmembrane topology map, which is this top figure here. So the substitutions were found to be distributed all the way through the VGSC. And these 4 homologous domains, D1, D2, D3, and D4. And this sort of like rolls up into a circle to make the pore. And the S5 and S6 segment of each of these domains are actually what makes the central pore itself that the ions move through. So we find SNPs all the way through, and we find a lot of SNPs in these 2 segments which make up the pore itself. And we can see that these are important. We know that L995, S, and F, they sit in one of these S6 linkers. So it's quite interesting that we find other mutations actually within the pore itself. And that's something that we will look at in a little bit more detail. So the 2 known resistance alleles affecting 995, F, and S had the highest overall allele frequencies, as we might expect. And these are very well known, very widespread KDR mutations. 995F was at high frequency in populations from West, Central, and Southern Africa. And 995S was at high frequency from populations in Central and East Africa. And that really fit the pattern that we were expecting from some previous research. What was quite interesting, we did find quite a few individuals in some countries that were heterozygous. So they carried 995F on one chromosome and 995S on the other chromosome. And we did wonder whether there might be some sort of selection for carrying both 995F and 995S. So we calculated p-values. So basically, whether we see these heterozygotes more than we would expect, given the number of homozygotes and heterozygotes that we find in those populations. And the results were interesting. In Cameroon, where we found quite a lot of these 995F and S heterozygotes, it didn't look to be significant. We didn't look to like we were getting an excess of 995S F heterozygotes. But in Gabon, it did look like there might be some selection. It did look like we were seeing these heterozygotes more than we expected. So potentially in some situations, there might be a fitness advantage for carrying both 995S and 995S, F and S. We also saw quite high frequencies of N1570Y. So this is this other known mutation that is found in concert with 995F and increases the insecticide resistance phenotype quite a lot. And we found that in Guinea and Ghana, Burkina Faso, Cameroon. But we were expecting to see that. That was something that we were aware of. Let's see what else you want to say about that.
Yes, I don't know if you're using your pointer, but we can't see a pointer.
Okay, well, yes, I've got two screens going here. So let me just go to the next.
So now we can see a pointer.
Cool. So let me see if I can fit this on the screen. Okay, so we had this abundance of high frequency non-synonymous variants. And so the next thing we looked at was linkage disequilibrium. So this is basically, it's just asking, do we see an unexpected association between different mutations? Do we see them together more than we would expect? So if we look at this matrix here, these are our 23 non-synonymous mutations on the X and the Y. And so each of these squares is a pairwise relationship between one of these mutations and another one of these mutations. And if we look at the scale on the right-hand side, if they've got a linkage disequilibrium of 1, dark blue, this means that when you see one of those mutations, you always see the other one. So they are always found together on the same chromosome. And if it is 0, if it's -1 or white, then you never see those mutations together. And what was really striking about this huge number of non-synonymous mutations that we just weren't expecting to see was that almost all of these new mutations are found exclusively on a 995F genomic background. So we've got this essential nervous system gene where we would expect that any non-synonymous mutation would be deleterious. It would be bad for the mosquito because it would change how this, it could change how the protein worked, and that might make the mosquito quite unhappy. 995S and F, we know, does have a fitness cost for the mosquito, but having a resistance phenotype just causes so much added fitness, so much benefit to the mosquito that it carries this mutation despite the fitness cost. So it was very strange to first find so many non-synonymous mutations and to find them on a particular background that already carried this KDR mutation. So what else have we got here? So we can see things that we expected, for example, on this plot. So N1570Y, we expect to always be on a 995F background. And there we can see it is. It's basically almost 100% of the time you find 995F with 1570Y. And then we find all these other mutations also only with 995F. Something else that was interesting here, there was a couple of mutations that we'd never seen before in Anopheles gambiae, which is these V402. So there's actually 2 nucleotide changes here that both cause the same amino acid change. They are in very high linkage disequilibrium with this other mutation, 1527T. So that's really interesting because 1527 occurs within a transmembrane segment that is immediately adjacent to residues with a predicted binding site for pyrethroid molecules. So it's plausible that a mutation at 1527 could alter this pyrethroid binding in the same way that KDR alters pyrethroid binding in these known KDR mutations. The 2 402 mutations that are in strong linkage with 1527 have been found in a number of other insect species and actually have already been linked and shown experimentally to confer pyrethroid resistance. So the fact that we're finding this haplotype, this on a chromosome, we're finding both 1527, which is next to a pyrethroid binding site, and 402 that has been shown in other insects to cause pyrethroid resistance really makes us concerned that what we have found here is another kind of pyrethroid resistance that are in these mosquito populations. What's interesting in this paper, when we wrote this paper, and I'll read to you what we wrote because it's quite interesting how wrong we were. So the species and geographical distribution of 1527 and 402 suggest they arose in West Africa in coluzzii and had not spread to other regions or to Anopheles gambiae at the time of sampling. The 1527 allele was present at a lower frequency than 995F, which is the known KDR mutation, in all of the West African Anopheles coluzzii populations. As 995F is known to have increased in frequency in West African coluzzii, we know that these KDR mutations are increasing and the pyrethroid resistance is increasing. What we suggested is happening here is perhaps this 1527 plus 402 combination of mutations. Perhaps it was an older pyrethroid resistance haplotype and is being replaced by 995F. Since we did this research, which is really great, based on this research, actually, based on these findings, a group in Liverpool went away and actually inserted these mutations into a susceptible mosquito population using like CRISPR techniques. They weren't able to get in 1527 and 402. I think they managed to just get in 402 into the susceptible population. And they actually found that 402 does confer pyrethroid resistance. And not only that, but it doesn't have the same fitness cost. So the larvae are fitter, more mosquitoes are closed, more eggs are laid than mosquitoes carrying 995F. So although it has a slightly less strong resistance phenotype, it actually has no detectable fitness cost. And in later data sets that we are analyzing now, we actually see that this 1527 plus 402 haplotype is rising in frequency. So it's really cool. This paper only came out in 2021. And since then, groups have, based on this research, gone away, validated 402, found that it is an important KDR or pyrethroid resistance SNP, and that it looks like it's increasing in frequency. So it's of even more concern than we had predicted in this paper. So let's move on to the next figure here. So I might try and make this a bit bigger.
There are a few questions that have popped up in the chat.
Okay.
I can read them too if you can.
Yeah, that's probably, if you ask me the questions.
Just because it's timely for what you were just talking about. Lucas asks, does the L995 background differ between countries? And Elijah asks, what are the known fitness costs of the L995 mutations to the mosquito?
So to answer the first question, we will get to that very shortly, and I will show a nice map figure that will answer your question. So what are the 995 fitness costs? That's a very good question. I might need to go back and read the paper, but it's things like, number of larvae, fitness of larvae, how many larvae pupate, how many pupae are closed, how many eggs are laid. There's a whole load of sort of fitness measurements that can be made and are made. I'll try and find a link to the paper and maybe I can email it round to everyone after we've finished. These measurements if you get a susceptible mosquito population and you insert 995F and you get a different susceptible mosquito population, you insert 402, you don't see those fitness costs in the 402 mosquitoes. So yeah, it's quite interesting. And there's been studies in other mosquitoes, I think in Aedes and things, looking at the KDR mutations in Aedes, and looking at fitness costs, and other fitness costs have been found caused by 995 type mutations. But yes, I will send a link round to everyone with that paper because it's really interesting. Right, so the next section is genetic backgrounds carrying resistance alleles. So the AG1000G data resource provides a rich source of information about the spread of insecticide resistance alleles, because data is not just available for the SNPs in the coding regions, but also in the flanking regions around the genes and in other genes. So these additional variants can be used to analyze the genetic background, and we call these haplotypes, on which resistance alleles are found. So here what we've done is we have used 2,284 haplotypes. So that's, we've got 1,142 mosquitoes, each with 2 chromosomes, one from the mum, one from the dad. We phased those into haplotypes. And then we've used hierarchical clustering, have we? What have we done here? No. So here we've used a median joining network to, where have I lost it? Yeah. So median joining network here allows for the reconstruction and placement of intermediate haplotypes that might not be observed in the data. We can see those on the figure here. Anywhere you see like a tiny black dot, this is where we've got an individual here, for example, that is 1 mutation away from this sort of central node. And then we've got another individual here that's 3 mutations away. Well, 1, 2, yeah, 3 mutations away from the central node. We don't actually have an individual here. But by using a median joining network, we can see that probably we just haven't sampled enough mosquitoes. We are assuming that probably these individuals exist or have existed to get from here to here. And it allows you to see those gaps potentially in your sampling. It also allows for non-hierarchical relationships, which might arise if recombination events have occurred between haplotypes. It shows you here where you've got different potential routes to get the same effect, where it's not clear which direction the network has gone in, but we certainly know that all these samples have these mutations. We reconstructed the network up to a maximum edge distance of 2 SNP differences. So anything further than 2 SNPs away that we don't have, we cut off the network. We've ended up here with these 10 median joining networks. It's quite neat because we get 5 which carry 995F, and we get 5 which carry 995S. And that really sort of suggests to us that there could well be 5 origins in our data set for 995F and 5 origins for 995S. So it looks like these mutations are arising multiple times in different populations and rising to high frequency because they're selected because of their insecticide resistance phenotype. The other thing that's kind of quite striking from this figure is what's going on with this F1 haplotype. And this goes back to what we were looking at with the LD matrix. It shows this really explosive radiation of amino acid substitutions secondary to the 995F allele. So within this F1 group, nodes carrying non-synonymous mutations, and these are shown by red arrows. I hope you can see that. They're quite fine. They're also labeled. So anytime you see a label, that's a non-synonymous mutation. So they all radiate out from this central node, which we presume is the ancestral F1 haplotype, which initially came under selection. So 995F appeared on a haplotype that gave an insecticide resistance phenotype. It rose in frequency, it was selected, but then lots of these non-synonymous mutations have occurred on that background. the suggestion there is, because these have then risen in frequency, that these are also under selection, and perhaps offer some sort of functional role. Perhaps they either increase insecticide resistance, or, as we've just learned about the fitness cost of 995F, it might be that these mutations are actually in some way ameliorating that fitness cost, so that the mosquito can be both insecticide resistant, but not carry any of the fitness cost from carrying that 995F allele. What's really interesting is we don't see that in 995S. We're not sure why that is. It might be that carrying 995S doesn't give you so much fitness cost. It'd be really interesting to see when groups using CRISPR or similar technologies put each of these mutations individually into susceptible backgrounds and measure the fitness costs for all of these one at a time. Then we'll really start to understand what's going on here. What else have we got next? So let's go to here, and this answers one of those questions. So we've got these 10 haplogroups, if you like, 5 very similar haplotypes containing 995S, 5 containing 995S. I've said F and S so many times, they're all starting to sound the same to me. So sorry if I'm saying the same letter over and over again. So we can look at a map, and we can plot these different haplogroups on a map, and we can see that there is indeed sort of a geographical spread to these different haplotype groups. In the far west, in Guinea-Bissau and the Gambia, we don't see any 995 alleles at all. It does look like there are other kinds of resistance there. We do find cytochrome P450 amplifications, and we'll be talking more about that kind of resistance in the next workshop. But as we move further east, we just see lots and lots of this F1 haplotype. That's this big group here, this big sort of explosive radiation here. We find that all the way sort of from Burkina Faso, all the way down to Angola, and we see that at quite high frequencies. In Gabon, we see some sort of quite interesting stuff that doesn't really look like this West African haplotypes. East Africa, we see a lot of this S3. Uganda's got a haplogroup that it's sort of private to that. And what's really striking here is what's going on in Cameroon in the middle. Now, the size of these points, if you like, is the number of haplotypes we have. So Cameroon does have the biggest sample set, but it carries almost every single haplotype group. So it's really interesting what's happening in Central Africa. In our new data sets, we actually do have samples from Central African Republic, from DRC, and we're seeing a similar pattern. So we're finding a huge amount of diversity of haplotype groups of carrying 995S or F in Central Africa, and we're not really sure why that is, whether these haplotype groups evolve in Central Africa and then move. I mean, we can see from how far this F1 haplotype group has moved that selection on these haplotypes is very strong. It's basically this is identical to haplotypes over here. So that's thousands of miles that these can travel. So have these all started in Central Africa and spread out, or somehow have these haplotypes all converged in Central Africa? And why does Central Africa hold so many of them at once? Why has one of these haplotypes not risen to the top as being the fittest and the other ones dropped out of the population? So this is a very interesting figure, and it raises a lot of questions that we don't have answers to at the moment, but it's pretty cool. Right, what's next? Yes, positive selection. So let's look at that. So to investigate for positive selection or non-synonymous alleles, we also performed an extended haplotype homozygosity analysis. It's called EHH for short. Basically, haplotypes under recent positive selection will have increased rapidly in frequency, and consequently, they'll have had less time to be broken down by recombination. And so they should have longer regions of haplotype homozygosity. So if you've got all of the haplotypes in the F1 haplotype group and line them up next to each other, you would expect them to be similar across their length for longer than haplotypes that don't carry a selected allele, because they'll have been recombining around for years and years and years, whereas these F1 haplotypes will have all risen to high frequency so quickly they won't have had time to break down through recombination. So we defined a core region, which is in the middle here around our VGSC 995 amino acid. And then we looked out in either direction from there, around 6,000 base pairs of flanking sequence. We were able to detect our 10 groups, haplotype groups, F1 to F5 and S1 to S5. And as expected, haplotypes carrying these 995F and 995S resistance alleles experienced a slower decay of EHH relative to wild type haplotypes. So we can see that here, where these, there's a pointer, here in the fine black lines here, wild type haplotypes, and we can see around our sort of central VGSC point, they drop very quickly down towards 0. And that's because they're just recombining quite naturally. They've been there for a long time. These thicker colored lines here are our haplotype groups. And we can see that they recombine, they're breaking down more slowly, particularly on this side. They're breaking down much more slowly than this side. That's actually because the centromere of the chromosome is here. And that actually sort of physically inhibits recombination to an extent. So recombination is always going to be slower close to a centromere. And I think that's just the effect you're seeing here, which is why you've got a much slower breakdown on this side. But still, on either side, it breaks down much more slowly than wild type haplotypes. We also found these two other haplotypes that seem to have this extended haplotype homozygosity. And one of those is this 402.15.27 haplotype, these pair of mutations in this haplotype group. So that was really interesting. And that's in the, I think that's this, which one is which? So that's this L1. Yes, this is why. So although we've got this nice extended haplotype homozygosity of this 402 plus 1527 haplotype group on this side of the gene, it breaks down very quickly on the centromere side. Now, I know I just said you don't expect very much recombination to happen on the centromeric side, but something has happened here that's caused this breakdown very quickly, and there's almost no extended haplotype homozygosity on that side of the gene. But actually, if you remember, there are two 402 mutations. There are two nucleotide changes that both cause the 402 amino acid change. And the reason that you're seeing this quick drop on this side is actually because you're looking at two different haplotypes on that side, one containing 402 from one haplotype and one containing 402 from a different haplotype that have recombined onto this 1527 background, which is why we see 1527 plus 402, but there are two versions of it. So I think that explains this, which so it still looks like this 1527 plus 402 haplotype group is under selection. It's just got this recombination event to bring the two 402s onto the 1527 background. So what else do we say about this? Yeah, so I think that's probably covered the different analyses that we did for this paper. Have we got any questions on that before I go on to the discussion? Is there anything there, Brandy, that would be good to discuss, or should we save it to the end?
There's a question that maybe you could think about more, and we could talk about later. Basically, how do we find out where different haplotypes come from? What have you done to try to answer that question? And if you haven't been able to, what sort of data might you need for that?
Yes, that's a very good question. And it's something that we're very interested in, and I have been thinking about for a number of years. The big problem with that, so what you need to do that really, you can look at a map and you can see that you've got haplotypes shared. We can go back to the map and have a look. So if we think about F1, a good question would be, where did F1 come from? Now, if you had a huge amount of sampling over the years, then you might be able to see it move. So, you know, it wouldn't be in this country and then it would appear in this country and then it would appear in the next country and so on. And you could trace it back and kind of work out where it came from. Unfortunately, we don't have that kind of sampling. We are now collecting those kind of data, but particularly for 995F, you know, we've missed that opportunity because it's spread so widely. So the next thing you would do would be to look, if you haven't got the actual sampling that's useful, you could look at the haplotypes, and you could look at how many sort of additional changes you find on these haplotypes, and that might show you whether you've got an older haplotype or a younger haplotype, and that might give you a clue as to where it's moving from, where it came from, the direction of travel. The problem there is we have very few wild type haplotypes. So in our populations, in our countries, in our samples, in our cohorts where we find 995S or 995F, we have very few wild type individuals. So when you're trying to compare the changes against a wild type, so, you know, first there will have been this 995F mutation, and then perhaps you'll find sort of other SNPs, synonymous or non-synonymous SNPs that you might be able to use to age that haplotype. We just don't have very much power because we don't have very many wild type samples, because insecticide resistance is so widespread, particularly caused by 995S and F. So there's not a really satisfactory, it's not a really great answer. Basically, as we get a load more data, and as we get a lot better sampling, particularly over time and also over space, I think we can begin to sort of brute force answer these questions, because we'll be able to see those movements in sort of real time. And that hopefully we'll be able to use that kind of information to look back in time, and kind of give us a clue for where things are happening, when they've happened, how they've moved, how fast they've moved, because that's really important to know, how quickly these mutations can spread. So yeah, it's a very interesting question, and yeah, hopefully one day we can get the answer to that. Right, so let's just quickly go through the discussion, see if there's interesting things here. So so what have we got? Cross resistance between pyrethroids and DDT. So this is quite interesting. VGSC is the physical target for both pyrethroid and DDT insecticides. And 995F and 995S both increase resistance to these insecticide classes. So some people have suggested that, you know, DDT might, these mutations might have been caused by DDT, and are being maintained by pyrethroid use, because DDT was used in Africa in the 1950s and 60s to eradicate malaria. It was actually used agriculturally before that, so it's been used for a long time. But hasn't really been used in a big way since then. Whereas pyrethroids are being used all the time, and all the bed nets are pyrethroid impregnated. So where's the question? So it was suggested that 995S, because it offers slightly more resistance to DDT than 995S, that must have been created by DDT. That's what drove that selective sweep. So if that was the case, then the 995S haplotype would be much older than 995F haplotypes. And our EHH analysis shows that that's not really the case. There's no significant differences overall between between the sort of the length of our 995F and the length of our 995S haplotypes. So it doesn't look like one of them is much older. So either they were both sort of initially selected by DDT, or they're both just being selected by pyrethroid resistant. But it's difficult to pick that apart. What else have we got? Resistance phenotypes for novel non-synonymous variants. So this is all quite complicated, and it's referring back to this figure right at the beginning. It's referring back to this figure here, which was done by a very clever chap at Liverpool, John Moores, called Andreas O'Reilly. And he's a protein person. So basically what he did was look at how the VGSC protein folds back in on itself. Because if you look at this naive diagram here, this is just sort of the pore spread out. We can see where these mutations are. And we can see, for example, that there's a load of mutations in the C terminus here, which doesn't seem to, it doesn't look like it's interacting with anything in this figure. But once you do these protein folding analyses, you can actually see that some of these mutations actually fall very close to important bits of the gene, to pyrethroid binding sites and things like that. So let's see so what was interesting here. Yeah, so for example, it shows the 402 residue is located in a very similar position on a different subunit to 995S. So that makes it look like it might be concerning in terms of what it does. It also showed that tail, which actually folds back in, that C-terminal tail folds back in. And so we found a lot of mutations in there, including 1874. We find a couple of mutations at that amino acid. And in some crop pest moths, that has been associated with insecticide, with pyrethroid resistance. And when we looked initially, we thought, well, it's right at the end. It's not doing anything important in this gene, so we're not sure how it might affect insecticide resistance. But Andreas's work shows that tail folds actually into the pore and actually could cause some problems like insecticide resistance. Okay, so the next thing that's sort of the final thing here, and possibly the most important, is a design of genetic assays for surveillance of pyrethroid resistance. Because what we have established here is the picture of insecticide resistance, of pyrethroid resistance, is much more complicated than just 995S or F. But yet those are what is being assayed in the field. That's what, is 995S or 995F present? If so, there's pyrethroid resistance in the population. But looking at those non-synonymous SNPs, looking at those, the EHH analysis, there's clearly a lot more complexity there. There's a lot more non-synonymous mutations that are involved, that are under selection. And there are even new combinations, new to us, 402 and 1527, that look like they have some important effect when it comes to insecticide resistance. So we need to be able to create insecticide resistance assays for the surveillance of this in a way that is, is cheap, is high throughput. It would be amazing to be able to just genome sequence everything, and that is coming down in cost. But it's just not practical. It's not practical, and it's not really necessary to sequence every single mosquito's genome. So to achieve substantial and spatial temporal coverage of mosquito populations, we wanted to be able to develop some sort of assay using technologies like amplicon sequencing. And in fact, some of this has already been put into practice. Our colleague Eric Lucas at LSTM has actually built an amplicon assay that enables you to very quickly look at thousands of mosquitoes at tens or hundreds of positions and sort of detect all these different non-synonymous alleles. And to do that, we actually used a decision tree. So we looked upstream and downstream in our haplotype groups, and we found all of the mutations, not just the non-synonymous mutations, but just all the differences between our different haplotype groups. And we asked our decision tree, how many SNPs do we need to be able to discern our 10 haplotype groups? So F1 to F5, S1 to F5. How many SNPs would you need in an assay to be able to determine what haplotype groups you have in your samples? And actually, we tried a couple of different methods, and we found, so it's when this let me find the figure, sorry. It's when this peaks, when this cross-validation score peaks, that's when you are able to discern all of your 10 haplotype groups. And we can see here it's about 20 SNPs. And these are two different methods, and they both sort of show a very similar thing. So to be able to pull apart our 10 haplotype groups that we have identified in these analyses, you need to be able to assay at least 20 SNPs. And so we have identified those SNPs. And actually, as part of this research, we have released supplementary data which contains the SNPs you need to identify the different haplotype groups, as well as all the SNPs upstream and downstream if you wanted to create your own primers and you needed to know all the different variation. So I think we're almost about run out of time, and it would be good to get some questions in. So I'm going to stop there. But yeah, I hope that was useful. I'm going to stop sharing my screen. I hope that was useful. And what questions do we have?
So Rosine asks, did you compare any population structure between wild type from GM and GW populations to other wild type in areas with high resistance allele frequency?
So again, the problem is that we have so few wild type in most populations that it's very difficult. You do see geographical differences between them. So the wild types in Gambia and Guinea-Bissau will look different to mosquito wild types from other bits of Africa. But it's very difficult to make a good comparison just because it's so hard to get hold of wild type mosquitoes.
And then there are a few comments about how far mosquitoes move. Do you have any comments based on these data about that?
Well, because we don't know the time scale. So there's a lot of research that looks at dispersal. There are groups, Tovi Lehman's group have been looking at long distance dispersal for a number of years. It looks like some mosquitoes can fly up very high and travel hundreds or thousands of miles. Whether that affects population, so population structure isn't clear. But I think we're more looking over a time scale of sort of 10, 15, 20 years. And I think probably for a lot of this, that's just what that movement looks like. That generation after generation, these genes can travel over quite some distance. But yeah, there's a whole group of people looking at actually how far can a mosquito travel, or how far can a gene travel, how quickly can a gene travel, because that's really the important question.
Yeah, so worth mentioning that those studies of distribution are extremely complicated and hard to do. So we are out of time, but please, anyone who has questions, you can post them in our Slack channel and we can get back to you. And of course, we have the workshop coming up, so that's a good chance to talk about anything that popped up here. Thank you very much, Chris, and thank you all for being here, and see you next week.
Thanks. Bye.
Bye.
Bye-bye.
Thanks, Chris.
No worries.
Thank you. Bye.
Okay, thank you, guys.
Thank you. Thank you, Chris.
Thank you, Chris, and everyone. Have a good day.
Thank you. Bye.
Bye.
Thank you.